It's not, you only get a little bit of ATP, so the purpose is not to make ATP. What could we say the purpose is?
Make electrons or to carry them to the
It's okay. So he's saying making these electrons, to carry the electrons, which these are electron carriers, NADH and FADH2, and to carry those electrons to this part here, which is the electron transport chain, okay? And so that's exactly right. So that's what the purpose of the Krebs cycle is. So I'm going to display the answers here, and we're going to go over this. I'm recording this for people who are drawing. So let's look at this. So here are your Krebs, this is the Krebs cycle. Remember, you don't need to know the intermediates here in the Krebs cycle. But where does it take place? As we drew on the board, it's in the matrix of the mitochondria. So you need to have more than just inside the mitochondria. So it has to be in the matrix. 17, what molecules introduced from the link reaction? And that is acetyl-CoA. Then it asks you, I had a lot of questions on this, if oxygen is needed as a reactant in the Krebs cycle. And the answer there is no. And how I can tell that looking by this diagram is there's no O2 going into it. So there's no arrow with O2 that's going in there. So that tells me that oxygen is not being used at all. So far, looking on the board, glycolysis, the link reaction, and the Krebs cycle, oxygen is not used in any of those reactions. So then 19 says, compare the oxaloacetate molecule with citrate. How many carbon atoms are in oxaloacetate? So you have to literally count them. So there are 4. And in citrate, if you count them, there are 6. So then there's a difference. Remember, and you can't just automatically just get 2 more carbon atoms. So where did they come from? And they came from the acetyl group, from the acetyl-CoA. And so that has 2 carbons here. The coenzyme A doesn't go into the citrate, and so it's from the acetyl group from that. All right, so then the next question asks you compare citrate with alpha-ketoglutarate. So citrate is a 6-carbon molecule, but alpha-ketoglutarate is only a 5-carbon molecule. So why is there a difference? Because 1 CO2 is released, so 1 carbon atom came off of that molecule. For the next one, alpha-ketoglutarate to succinate, this is a 5-carbon molecule. Now we go down to a 4-carbon molecule. Where did the extra carbon go? Carbon dioxide. All right, and so we see a pattern there. So that was 20 and 21, what I just answered there. So then 22 asks you energy is transferred to other high energy potential energy molecules. List those molecules and indicate the number produced. So therefore, in the number produced, it says with 1 turn of the cycle. So you get 1, 2, 3 high energy NADHs. You get 1 FADH2, and you get 1 ATP for 1 turn of the cycle. All right, so that's what you should have for 22. 23 and 24 I had a lot of questions on because it asked you, out of these 4 intermediates in the Krebs cycle, which one has the highest potential energy? And so I had a lot of people come up to me and say, how do I know? What am I looking for to tell if it has high potential energy? So what you have to do is look at the reactions. So let's look at citrate being converted to alpha-ketoglutarate. So when we look at this here, during that reaction, what this shows is that NAD plus is becoming NADH. This is a reaction that needs energy. You're putting things together, you're building NADH, and so this needs energy. And so therefore, where does that energy come from? It comes from the breakdown of citrate into, the conversion of citrate into alpha-ketoglutarate. So this right here, process releases energy, and some of that energy now goes to make NADH. So some of the energy that was once in citrate is now in NADH, and so therefore alpha-ketoglutarate has less energy than citrate because some of the energy is now in the NADH molecule. And as we go along, alpha-ketoglutarate going to succinate, same thing. Some of the energy in alpha-ketoglutarate goes to making NADH and ATP, so succinate has less energy than that. And then the same thing between succinate and oxaloacetate. So therefore, when looking at this, the one that has the highest potential energy would be the one here at the beginning, which is citrate. And then the next question I want to ask you, which one is the lowest, that would be oxaloacetate. all right, because a lot of that energy went to making these other high energy molecules. And then lastly, number 25, what can you conclude about the potential energy of the acetyl group that's carried from the link reaction to the Krebs cycle? And does this group of atoms have a high potential energy or a low potential energy? So the acetyl group comes and reacts with this low energy. We said that this has the lower potential energy. Acetyl group reacts with oxaloacetate to form citrate. And we just said that citrate is a high energy molecule. So therefore the acetyl group must have a high potential energy. All right? And so that's the answers there. All right? And so then 26, how many terms of the Krebs cycle is needed for every glucose? Remember, that's 2. And then the 27, your totals, you should have 4 CO2s are produced. You should have 2 ATPs. You should have 6 NADHs and 2 FADH2s. This is for both terms. And lastly, was oxygen used as a reaction for glycolysis link or the Krebs cycle? And no, it has not been. All right? And so I'm going to hold off on going over the extension questions until we continue going over the big picture here until we finish this process of cellular respiration. All right, so we'll turn our attention here to this process. And so we left off yesterday, so we said that and this last step is where the majority of ATP is going to be produced. And so, let me fix my. Majority of ATP is going to be produced. And so, we said that the NADH, which is an electron carrier, comes to the electron transport chain and drops off its 2 electrons and moves through that electron transport chain and eventually meets up with hydrogen and ions to make hydrogen atoms to form water. So that is why water right here is a product of cellular respiration, and oxygen is a reactant. So the oxygen that is going to become part of this water here. Now, what did we say? This is where we left off with. What did we say when the electrons move through the electron transport chain? What did they do? And I drew it in here yesterday. I left this up from, and I don't have it here on there. What are the? I'll have to redraw it here. What do the electrons do as they're moving through?
Well, for the amino acids, they first cause them to become reduced and oxidized, and then for the transport proteins, for the proteins, they energize them, I believe, and they cause them to go through active transport to where they pump hydrogen ions outside of the inner membrane.
Mm-hmm. And so they pump it outside in the intermembrane space right here. And so I'm going to redraw this. What color did I do the H pluses in this class? Green? Blue? All right, so people, maybe, I'll since I made these blue, every hour I just grab the color so it's a little bit different here. I'll just draw them here with green here. H plus, all right, goes out here. Well, let's try it here. So the H plus is moving this way, all right, being pumped. Pumped meaning it requires energy, right? And so the energy comes from the redox reactions of these electrons being passed from molecule to molecule because each one is more electronegative than the next. So I think this is where we stopped yesterday. I think I posed the question yesterday in terms of why is it important that these hydrogen ions be pumped here. What this is doing is creating a really high concentration of H pluses in this space. And it's pumped, meaning it goes from low to high concentration. So we're making it from low to high concentration, making this concentration even higher. What purpose is having a high H plus concentration in this space here? What's the purpose of that in cellular respiration? That's the question I left off with you yesterday. Why would the cell want to do that? Grace?
Is it to reduce the oxygen
All right, so it does use the oxygen and produce water, so they do, but the H plus is out here, the water is made here, so the H pluses are in this space here. So what is the H pluses right here? Why do we want a high concentration of them here? Give you a hint, do they stay here? They do not stay here. Can somebody tell me where they go? Where do they go? What do you think? Peter?
They go into the intermembrane space.
That's where they're at. I'm asking you from
and they go
there.
back into the matrix.
They go back into the matrix. How do they do that?
ATP synthase.
Nice. All right, ATP synthase. And so ATP synthase is another molecule that's completely different than the electron transport chain. So this part right here, let's put here, this is the ETC, this is the electron transport chain. So now we have another molecule, completely different, and let's see, I don't have very many colors. If I had another color, I'd make it completely different, but I wrote here, so I'm going to make this ATP synthase. Let's draw it right here in the, all right, so this is ATP synthase. It's totally a different molecule. Ends in ASE. ASE means what when something ends in ASE? Liam. It means it's an enzyme. So enzymes can be within the membrane and be a protein within the membrane. So, and it's called ATP synthase because synth is coming from synthesis, so it's going to make ATP, which remember I said this last step is where you make the majority of ATP. We've only made 2 and 2 here, so that we've made 4 so far, not a lot of ATP. So the majority of ATP is going to be made here. And so as Peter said that these H pluses are going to diffuse back in, all right, like this, diffuse back in to the matrix of the mitochondria through this ATP synthase. This is really important, this important step here. Why is that important? The reason why is because before the H plus is diffused through, that ATP synthase is not in the right shape. And so remember, shape for enzymes are very, very important. That the active site has to be in the right shape for the substrates to bind to it. Can somebody tell me ATP synthase, we're going to make ATP, what 2 molecules are put together to make ATP? What 2 things are put together to make ATP? What do you think? Betsy, what do you think? What 2 molecules are used to make ATP? What are we going to hook together to make ATP? All right. What do you think, Luke? ADP and what are you going to hook to it? To make a phosphate group. All right. So those are your 2 substrates. So what happens is ADP and the phosphate can't bind to the active site of ATP synthase unless it's in the right shape. So H plus is diffusing through there, changes the shape of the enzyme, and when the enzyme shape is changed, then the ADP and the inorganic phosphate, ADP and the phosphate, can come and bind to the active site and ATP can be made. So ATP is made there. And it's only when those H pluses are diffusing through. Hence the reason why it's so important to have a high concentration of H pluses in the intermembrane space. If these guys, H pluses, ever reached equilibrium, organisms would die very, very soon because the H pluses would never diffuse back in, and then this enzyme wouldn't work, and therefore we wouldn't be able to make ATP. All right? And so, so therefore, what happens is, for each NADH molecule, when it drops off the 2 electrons, and the electrons go through the electron transport chain and end up with water, each one pumps 3 H pluses into the intermembrane space, and then each H plus diffuses down independently, so every NADH makes 3 ATP. You see that? Each NADH drops off the electrons, pumps 1, 2, 3 H pluses. So 1, 2, 3 H pluses diffuse through, 3 ATPs are made. So I talked about that in the notes, that 1 NADH makes 3 ATP. All right, there was one other thing, one other electron carrier. What's the other electron carrier? Grace? FADH2, and FADH2 is coming here only from the Krebs cycle, and there's only a couple of them. And these guys are unique in that they don't drop off the electrons at the same place that NADH drops off the electrons. Do you remember where they drop them off at? Sam?
Is it just later in the electron transfer chain compared to NADH?
Yeah, later in the electron transport chain. So if you recall, I'm going to show you the picture from the notes to just kind of make sure that we remember from the notes here. This picture here, where here's your NADH dropping off at the beginning of the electron transport chain. FADH2 bypasses that first transport complex, and so drops it off here. And so let's draw this on our thing here. That I'm going to write here that here 2 electrons from FADH2 are dropped off later. All right. So it bypasses this first pumping of H pluses, so it'll be converted back to FAD. And these electrons, therefore, only go through 1, 2 of the complexes here. They still go to make water, but 1, 2. So therefore, FADH2 pumps 1, 2 H pluses, which means 1, 2 ATPs can be made. All right. So FADH2 makes less ATP. All right, this still helps to make ATP, but per molecule, less ATP there. All right, and so, so therefore, when we talked about this in the notes, we said that 1 NADH, remember, makes 3 ATP, so you get 30 there. And then what did we just say? 1 FADH2 makes what? 2. All right, 2 ATP. So we get an additional how many? 2. All right, and so so therefore, this is for this is for your totals here, and so so you get the majority of your ATP being made in this last step. Which, by the way, let's talk about names here, because some people first hour were asking about this, so I'm assuming you guys have questions here. This whole last step, remember we've been talking about glycolysis and the link reaction, the Krebs cycle. This whole last step is called oxidative phosphorylation. So if you look at this picture from the notes, it says oxidative phosphorylation. And there's 2 parts to that. There's electron transport and chemiosmosis. So the electron transport is the electron transport chain, which its goal is to make H pluses, pump the H pluses into the inner membrane space. And then the chemiosmosis part is when they diffuse back down. So I'm going to call this, so I said this is electron transport. This is chemiosmosis. And then these guys together are called oxidative phosphorylation. So you see the relationship there. And this should be 3. Did you guys notice that? Yeah. All right. I was getting ahead of myself because we're going to total it. It should be 3 ATP. 1 times 3 is not 30, if you weren't sure about that. All right. All right, so I was thinking ahead because total here, so let's talk about total ATP. How many NADH total come to the electron transport chain to drop off electrons? 10, right? So you have 6 from the Krebs cycle, 2 from the link reaction, and 2 from glycolysis, that gives us 10. So each NADH makes 3 ATPs, so 10 times 3, that's the 30 I was in my head. All right, thinking. And then FADH2, how many of those are there? 2 from the Krebs cycle, so therefore you get another 4. And so therefore, notice here it says about 32 or 34. And I want to spend a minute and talk about that range here. This is not an exact science here. In fact, this last step is still very much being researched currently. And so it's been researching ever since these numbers here and exactly how many H pluses go out and how many H pluses go in has changed periodically since I've been teaching. So they're still researching about that. So I just wanted to point that out, that science is always a work in progress, and this is something that they're still working on. In fact, they now think, so we're going to go with this for simplistic case here. I rounded 3 and 2. But they really think, in their current research, they really think that when the electrons go through for an NADH, that it's actually 10 hydrogens that get pumped here. So they're not sure exactly how many. It's not as clear cut as 1, 1, 1 or 2, 2, 2, because 10 is not easily divisible by 3. All right, so they think that 10 of them go in to the inner membrane space, and the current research is that they think how many have to go through the ATP synthase to make ATP. They currently think 4 of them. So it's not an exact number, it's not a nice round number. And so in our current textbook, so I rounded here, but in our current textbook, that what they actually think is that for every NADH molecule, that 2.5 ATPs on average are made, all right? So it's 2.5, and then for every FADH2, they think it's actually 1.5. So it's not an exact science. So therefore, in our textbook, it'll say, so this number is rounding for teaching purposes. I just wanted you to understand, it makes it easier to understand. This is the rounding number. In our current textbook, it says 28 to 30. ATP, and this is with no rounding. So this is the 1.5 and the 2. What I want you to understand is I just don't want you to be confused if you see 28 to 30 or 32 to 34. It depends upon if you're using exact numbers or rounding. And so I'm not going to ever ask you, and on the AP tests, they're not going to ask you how many ATPs are made by oxidative phosphorylation. You need to understand the big picture. The majority of ATP is made in this process. The number is not even agreeable among scientists. They're still researching it, all right? But either way, so it just depends on if you round it or not, okay? And so why is it a range? So why is it, say, 32 to 34 or 28 to 30? Why is it not at least rounding an exact number? And that has to do with a couple of things. NADH that's made in glycolysis has to come into the, to drop off its electrons, so the electrons have to come into the mitochondria. In researching that, they actually found out that NADH can't get through the mitochondria, all right? So therefore, this NADH in the glycolysis stays out in the cytoplasm, but what they found is that there are proteins that are electron shuttles that the NADH can actually give its electrons to. The electrons go through the membrane proteins, these shuttles, and meet up with an FAD molecule or an NAD plus molecule. And so depending upon which one they meet up with, and they enter in at different levels in the electron transport chain. If the electrons from NADH meet up with the FAD molecule and become FADH2, less ATP is made. If it meets up with an NAD plus molecule and it makes an NADH, more ATP is made. So therefore you have the lower end and the higher end there. And so that's the reason why. All right? And that is cellular respiration in a nutshell. Let me ask you this. The inner membrane has folds. So going back from when we talked about with the cells and surface area, so hopefully you'll be able to tell me that it has folds because it increases surface area. Can somebody tell me why having a lot of surface area of the inner membrane is important? Why is having a lot of surface area of this inner membrane important? And why isn't it important, the outer membrane's not folded, just the inner membrane? Why is that? What do you think, Hannah? To be more efficient. Why would it be more efficient? Somebody else tell me why it's that.
It has higher surface ratio, surface over volume ratio.
All right, high surface area to volume ratio. Why is that important? Why isn't it on the outside? Why does it have to be in the inner membrane that this is all important? Jesse?
Because the inner membrane has the electron transport chain, so that more ATPs
That's right. I just drew one electron transport chain and one ATP synthase in here. But realistically, there's going to be another electron transport chain, another ATP synthase, another electron transport, and another one, and so on and so forth. So the more inner membrane you have, the more electron transport chains and ATP synthase you can have, which means then the more ATP can be made. All right? And so therefore, that is why the inner membrane's folded. The outer membrane has nothing to do with cellular respiration. The outer membrane is just there to contain everything in the cytoplasm. So therefore, that does not need to be folded. All right, and so that is an advantage for that, and why we need high surface area. All right. Okay. So any questions on this? So let's answer those last few questions in your packet from yesterday. All right, so 29, identify two places in the Krebs cycle where a decrease in free energy is coupled with an increase in free energy. And so going back to this is just like that citrate to alpha ketoglutarate. That is a decrease in free energy, higher free energy to low. And that's coupled with this reaction where when you make NADH, it goes from low to high free energy. So some of that energy in the citrate molecule is now in the NADH molecule. So that would be an example there. And then 30, let's talk about 30 here in terms of this big picture. So 30 is talking about different things that we eat in our diet and how that relates to cellular respiration. So I started out with cellular respiration with glucose. And so here's the glucose and going all the way to the end and how many ATPs can be made, or adding up these guys here. And so it makes a lot of ATP. So when it's talking here about what we have in our body and what we can use for energy, glycogen is, remember, our body's way of storing excess glucose. So that happens in our liver and our muscle cells, where excess glucose from our diet, we put them in long chains, and then we break it down when we need it. So when glycogen is broken down, we get glucose, and glucose goes through the whole process of cellular respiration, and we get some ATP. So we can add up all these and get the ATP total. Let's talk about fat. Fat, if you recall, fat is a triglyceride, and again, I drew it in this chapter, like it looks like the capital letter E, a glycerol with 3 fatty acids attached to it. But when we eat a fat molecule, we digest it, and we digest it into its smaller molecules. And so what happens is, instead of one big molecule, we're going to end up with one glycerol here, and then the fatty acids are going to break off from the glycerol, and so we're going to end up with one glycerol and 3 fatty acids here. And so we end up with, instead of one big molecule, 4 smaller molecules. And each one of those 4 smaller molecules can actually go through cellular respiration and enter at different points, which is what this was telling you here. So the glycerol molecule, when we break it down, actually can be converted by our cells to pGal. So pGal here, where does pGal? It's in here in glycolysis. So if you look here, when it enters as pGal, that means that you can do the rest of cellular respiration. So actually, if you don't start as glucose and you start here, you actually make more ATP because you bypass the energy investment phase. Do you see that? All right. And so therefore, it goes through the whole process here, and we get all of this ATP. Then, that's only one part of the fat molecule. Remember, there are 3 other parts here, because we digested it now into 3 fatty acids. So that's why it says 3 fatty acids, and these guys get converted in our cells to acetyl-CoA. And notice it says the number varies, and that's because fatty acid length can vary. So you can have long fatty acids, short fatty acids. The longer fatty acids you have, the more acetyl-CoA you can make in your cells. So acetyl-CoA, where does that enter? That enters in here at the Krebs cycle. So now, how many acetyl-CoAs are you going to have? You're going to have at least 3, all right, for the 3 fatty acids, but it can be way more than that, depending on how big those fatty acids are. And so we know at least that when acetyl-CoA comes in, and then we go through the Krebs cycle and going through the rest of cellular respiration, we're going to get more energy, 32 to 34, and 2 here. So, and that happens multiple times because there's multiple fatty acids. And so which one has more, where are we going to get more ATP, a glucose or a fat molecule? Glucose or a fat molecule? What do you think? Caitlin?
A fat molecule.
A fat molecule, and it's a lot more because of it breaking down into those 4 different molecules entering at the various points throughout cellular respiration. And then lastly, proteins. Proteins, we can break down proteins to get energy. This is not ideal. So usually what your body will do is when you need energy, it will use your glycogen, and use your glucose storage first, and then it will start to use your fat storage, all right? So start to break down fat. We have special cells called adipose cells that store fat in our body. And it'll start to break down fat for energy. And then if you really are malnutrition and your cells need energy, it'll start to break down protein. And protein, when we break down protein in our body, it breaks down into amino acids, which can be converted to pyruvate and acetyl-CoA. So it can convert to here or here. So therefore, it can do the rest of cellular respiration so we can get energy from that. But that's not a very healthy thing to do because protein, the protein sources that are usually starting to break down are all your protein in your muscles. So your muscles will start to deteriorate and break down. And so that's what happens kind of when cells are starved. Question?
I was just wondering, for each Krebs cycle, does that mean that 3 NADH are produced instead of 6 total?
Yes. So because it turns twice for every glucose, so yes, 6 total. Yeah. Jesse, yours.
So like if there was like a bigger person and then like a skinnier person and then they both starve themselves, would like the bigger person last longer?
Yeah, like they would. Yes, it's actually an evolutionary advantage because way back in the day when we didn't have grocery stores to go to, we would have to go hunt and gather to get our food, and food sources were not always plentiful. So therefore, that storage is your body's way to be able to survive through times when food is not plentiful. The thing nowadays is that food is readily plentiful, and we have a lot of energy in those foods, and so we store them a lot, all right? And so therefore, like, you know, in the United States, the obesity problem is going up, all right, because of that. But yes, to answer your question.
What is urea?
What is what?
Urea.
Urea is a molecule that is released in your urine, all right? It's the part of urine from urine. All right, and so answering these questions, and I think I already answered most of them. So glucose that enters in glycolysis, P-gal is in the glycolysis, acetyl-CoA, the Krebs cycle, and pyruvate is the link reaction. For the next one, when I asked you, you should have that fat makes way more ATP. You don't have to calculate a number there for B, just that that fat will have more. And then letter C I just talked about, and that is the breaking down of muscle tissue. All right, for that. Okay. All right, so any questions on that? All right, so the two things that you picked up today, one is a new instruction sheet that goes with the new textbook. That's the online part of it, and so it's the same instruction sheet with new codes and so on. So I've had several people asking me to give the new instructions for the new textbook. So here they are. I do want to point out in the center there, it says you most like, in bold and underline, there's two things that are bold and underlined I want to point out. One that says you most likely do not have a Pearson account, so click no. If you do have a Pearson account, meaning that if you made an account for the old textbook, you have a Pearson account, so you're not going to click no. So then notice the or, and I underlined here. If you have a Pearson account, click yes, and it'll ask you to enter your current username and password, so then you don't have to re-register, all right? So I just wanted to point that out. These codes are for 50 students each. I have 94, so I have just enough codes. So again, if you use the first code and it doesn't let you, go to the second code. All right, it just means that 50 students have already signed in. And then everybody right now circle second hour, so that for the class down here, so that you put in the right class code and don't go into another class. All right. So I'm going to give you time right now to look at the second sheet, which says oxidative phosphorylation. So these are just some questions about this process that we just went over here to kind of finish up cellular respiration on the board. And so I'm going to give you the rest of the hour to work on those. Whatever you don't get done will be for homework. And I have one last little small video to finish up. If you notice, there's just a little bit left in our notes that we're going to wrap up. Okay? All right.